Plant kingdom. In the previous chapter, we looked at the broad classification of living organisms and the system proposed by Whitaker in 1969, wherein he suggested that 5 kingdom classification, that is Monera, Protista, Fungi, Animalia and Plantae. In this chapter, we will deal in detail with the further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of Monera and Protista having cell walls have now been excluded from Plantae, though earlier classifications placed them in same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe Plantae under algae, bryophyte, teratophyte, gymnosperm and angiosperms. Let us also look at the classification within angiosperm to understand some of the concerns that influenced the classification system. The earlier system of classification used only gross superficial morphological characters such as habitat, color, number and shape of leaves, etc. They were based mainly on vegetative characters or on the androecium structures system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and system sexual characters. This is not acceptable since we know that often the vegetative characters are most easily affected by the environment. As against this natural classification system developed, which were based on natural affinities among the organisms and considered not only the external features but also internal features like ultrastructure, anatomy, embryology and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. Bentham and Hooker classification. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assumes that organism belongs to the same taxa, have a common ancestors. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characters. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior and chemotaxonomy that use the chemical constitution of the plant to resolve confusion, are also used by taxonomists these days. Algae. Algae are chlorophyll bearing, simple, thalloid, autotrophic and largely aquatic, both freshwater and marine organisms. They occur in variety of other habitats, moist stones, soils and wood. Some of them also occur in association with the fungi, that is lichens and animals, example on sloth bear. The form and size of algae is highly variable. The size ranges from the microscopic unicellular form like Chlamydomonas to colonial forms like volvox and to the filamentous forms like Alothrix and Spirogyra. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by production of different types of spores, the most common being the zoospores. They are flagellated, motile and on germination gives rise to a new plant. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Chlamydomonas or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in some species of Chlamydomonas is termed as anisogamous. Fusion between gametes is termed oogamous. Example, fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. Example, volvox fucus. Algae are useful to man in variety of ways. At least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, They increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porphyra, Laminaria and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, water holding substance, example algin, brown algae and carrageen, red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella and Spirulina are unicellular algae rich in proteins and are used as food supplements even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. Chlorophyceae, the member of Chlorophyceae, are commonly called green algae. The plant body may be unicellular, colonial and filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain proteins besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type of formation of sex cells and it may be isogamous, anisogamous or oogamous. Some commonly found algae, green algae, are Chlamydomonas, Volvox, Ulothrix, Spirogyra and Chara. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show greater variation in size and forms. They range from simple branched filamentous forms, ectocarpus, to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminaria or mannitol. The vegetative cells have a cellulosic wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a hold force and has a stalk. The stipe and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in the most brown algae is by biflagellated zoospores that are pear-shaped and have two unequally laterally arranged flagella. Sexual reproduction may be isogamous, anisogamous or oogamous. Union of gametes may take place in water or within the oogamous species. The gametes are pyriform, pear-shaped and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum and Fucus. Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment or phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in warmer areas. They occur in both well-lighted regions close to surface of water and also at great depths in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organisms. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common members are Polysiphonia, Porphyra, Gracilaria and Gelidium. Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist, shady areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shady localities. They play an important role in plant succession on bare rocks or soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect, and attached to substratum by unicellular or multicellular rhizoids. They lack true roots, stems, or leaves. They may possess root-like, leaf-like, and stem-like structures. The main plant body of bryophyte is a halophyte. It produces gametes, hence is called a gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellated antherozoids. The female sex organ called archegonium is flask-shaped and produces a single egg. The antherozoids are released into water where they come in contact with the archegonium. An antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellulate body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nutrition from it. Some cells of the sporophyte undergo reduction division, that is meiosis, to produce haploid spores. These spores germinate to produce a gametophyte. Bryophytes in general are of little economical importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses, along with the lichens, are the first organisms to colonize rocks and hence are great ecological importance. They decompose rocks, making the substance suitable for growth of higher plants. Since mosses from dense mats on the soil, they produce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy grounds, damp soil, bark of trees, and deep in the woods. The plant body of the liverwort is thalloid, example Markantia. The thallus is dorsiventral and closely pressed to substrate. The leafy membrane have tiny leaf-like appendages in two rows on the stem-like structure. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structure called gemma, plural gemmae. Gemmae are green, multicellular, asexual buds, which develop in small receptacles called gemma cups located on the thalli, and gemma become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on same or on different thalli. The sporophyte is differentiated into a food, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Mosses. The predominant stage of life cycle of a moss is gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema of the lateral bud. They consist of upright, slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leaf shoots. After fertilization, the zygote develops into a sporophyte consisting of a food, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverwort. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore disposal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophytes. Pteridophytes include horsetails, ferns, Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess a vascular tissue, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase of the life cycle is gametophytic plant body. However, in pteridophyte, the main plant body is sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well differentiated vascular tissue. The level of pteridophyta are small. The leaves of pteridophyta are small microphyllous as in Selaginella or large macrophyllous as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structure called strobili or cones, Selaginella and equistem. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophyte called prothallus. These gametes require cool, damp, shady places to grow. Because of this specific restriction requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of antherozoids. The male gametes released from the antheridia to the mouth of archegonia. Fusion of male gametes with the egg present in the archegonia results in the formation of zygote. Zygote thereafter produce a multicellular, well-differentiated sporophyte, which is the dominant phase of pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro large and micro small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophyte respectively. The female gametophytes in these plants are retained on the parent sporophyte for variable periods. The development of zygote into young embryos take place within the female gametophyte. This event is a precursor to the seed habitat considered as an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, example Psilotum; Lycopsida, example Selaginella and Lycopodium; Spinopsida, example Equistem; and Pteropsida, example Dryopteris, Teres, and Adiatum. Gymnosperm. The gymnosperm are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, that is, are naked. Gymnosperm include medium-sized trees or tall trees and shrubs. One of the gymnosperm, the giant redwood tree Sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, pinus, while in some other cycles, small specialized roots called coraloid roots are associated with nitrogen-fixing cyanobacteria, maybe cyanobacteria. The stems are unbranched as in cycas or branched as in pinus, cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperm are well adapted to withstand extreme of temperature, humidity, and wind. In conifers, the needle-like leaf reduce the surface area. The thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are born on strobilus, that are born on sporophyll, which are arranged spirally along an form axis to form lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia. The strobili bearing microsporophylls and microsporangia are called microsporogenate or male strobili. The microspore develop into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cones bear megasporophyll with the ovules in ovules or megasporangia are called macrosporogenate or female strobili. The male or female cones or strobili may be born on same tree, example in Pinus. However, in Cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of nucleus of the nucellus. The nucellus is produced by envelopes and the composite structure is called ovule. The ovule are born on megasporophylls which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium, that is nucellus, develop into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm, the male and female gamete do not have an independent free-living existence. They remain within the sporangia, retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of ovules born on megasporophylls. The pollen tube carrying the male gametes grow towards the archegonia in the ovule and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and ovules into seeds. These seeds are not covered. Angiosperms. Unlike the gymnosperms, where the ovules are naked, in the angiosperms of flowering plants, the pollen grains and ovules are developed in specialized structure called flowers. The angiosperms, the seeds are enclosed by fruit. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from tiny, almost microscopic Wolffia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicine and several other commercially important products. They are divided into two classes, the dicotyledons and monocotyledons. The dicotyledons are characterized by having 2 cotyledons in their seeds, while the monocotyledons have only 1. The male sex organs in a flower is the stamen. Each stamen consists of a slender filament with the anther at the tip. The anther following meiosis produce pollen grains. The female sex organs in the flower is the pistil or the carpel. Pistil consists of an ovary enclosing one to many ovules. Within ovules are present highly reduced female gametophyte termed embryo sacs. The embryo sac formation is preceded by meiosis. Hence, each of the cells of an embryo sac is haploid. Each embryo sac has 3 cells. Example: 3 cells. Each of the embryo sacs has a 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuses to produce a diploid secondary nucleus. Pollen grain after dispersal from the anther are carried by winds or various other agents to stigma of the pistil. This term is pollination. The pollen grain germinate on the stigma and the resulting pollen tube grows through the tissue of stigma and style and reaches the ovule. The pollen tube enters the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cell to form a zygote, syngamy. The other male gamete fuses with the diploid secondary nucleus to produce the triploid primary endosperm nucleus. Because of the involvement of two fusions, this event is termed as double fertilization and even unique to angiosperms. The zygote develops into an embryo with one or two cotyledons, and the primary endosperm nucleus develops into endosperm, which provide nourishment to the developing embryo. The synergids and antibodies degenerate from after fertilization. During these events, the ovule develop into seeds and the ovaries develop into fruit. The life cycle of an angiosperm is shown in plant life cycle and alternation of generation. In plant, both the haploid and diploid cells can be divided by mitosis. This ability leads to formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophytic. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophyte. Plant body. The haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals represent them differ in following patterns. Number 1. Sporophytic generation is represented only by the one cell zygote. There are no free living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plants is the free living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, pyrogyra and some species of chlamydomonas represent this pattern. On the other hand, extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic. All seed-bearing plants, that is, the gymnosperm and angiosperm, follow this pattern. Third one is bryophytes and pteridophytes. Interestingly, exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however, they differ in their dominant phases. A dominant independent photosynthetic thalloid or rare phase is represented by a haploid gametophyte, and it alters with short-lived multicellular sporophyte. Totally or partially dependent on gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alters with the multicellular sporophytic or autotrophic, the independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them, example Ectocarpus, Polysiphonia, kelps, are haplodiplontic. Fucus and algae is diplontic.